Good morning, student. I am Mr. Vinod Thomre. We are studying plant physiology, and our today's topic of plant physiology is plant growth regulators. Plant growth regulators, they are the chemical substances that influence the growth and differentiation of plant cells, tissues, and organs. Plant growth regulators functions as a chemical messenger for intracellular communication. They work together coordinating the growth and development of cells. They could be indole compounds, adenine derivatives, or derivatives of carotenoids, or terpenes or gases. Phytohormones or plant growth regulators, they are of five types: auxin, gibberellins, cytokinins, abscisic acid, and ethylene. Those plant growth regulators, they are classified into two groups: growth promoters and growth inhibitors. In growth promoters, the plant growth regulators are auxin, gibberellins, and cytokinin, and the abscisic acid and ethylene, they come into the category of growth inhibitor. The first phytohormone we study is auxins. Auxins were discovered by Charles Darwin and Francis Darwin. They isolated auxin from the tip of coleoptile of oat seedling. Went to develop a Venn curvature test for bioassay of auxin. Here, synthetic auxins, example of synthetic auxins: indole acetic acid, indole butyric acid, naphthalene acetic acid, 2,4-dichlorophenolic acid. The physiological effect of auxin: it promotes apical dominance. Auxin initiate rooting in stem cutting. Stem cutting is a kind of vegetative propagation method. Promote flowering in case of pineapple. It prevents leaf drop and fruit drop at earlier stage. It promotes the abscission in case of older mature leaves and fruits. It induces parthenocarpy, for example, tomatoes. It may be used as a herbicides. It controls xylem differentiation and helps in cell division. The next plant growth regulator is cytokinin. Skoog and Miller discovered cytokinins. They crystallize the cytokinesis, promoting active substance named it kinetin from corn kernels and coconut milk. Cytokinins physiological effect in case of plants: it induces cell division, it delays the senescence of leaves and other organs, hence they enhance the life of leaves and other organs. They promote lateral bud formation. Cytokinin increases cell expansion in dicotyledons and in leaves. They promote chloroplast development and chlorophyll synthesis. The next plant growth regulator is gibberellins. A Japanese scientist identified gibberellins from the fungal pathogen called Gibberella fusicori. The gibberellins were isolated from infected rice seedling when treated with sterile filtrate of fungus. If we see the physiological effect of gibberellin, then it shows elongation of intact stem. That is, it helps in cell elongation. It causes dwarf shoots. Bolting in rosette plants, for example, henbane and cabbage. It overcomes natural dormancy of buds, tubers, seed. That means it helps in seed germination. It induces parthenocarpy or development of seedlet fruits. It promotes flowering in long day plants, and it also plays an important role in vernalization. That means the induction or promoting flowering by the treatment of cold temperature is called vernalization. The next plant growth regulator is ethylene, which is called growth inhibitor. Cuisine confirmed the release of volatile substance from ripened oranges that hasten the ripening of stored and ripe bananas. The volatile substance was identified as a ethylene. Let's see the physiological effect of ethylene. There is the small change. This is not the physiological effect of cytokinin. Those are the physiological effect of ethylene. Physiological, we are studying physiological effect of ethylene. Ethylene promotes senescence and abscission. As we know that ethylene is a stress hormone. It inhibits elongation of stem and roots. It induces fruit ripening by enhancing the respiration rate in the wall of fruit. It breaks seed and bud dormancy. That means it helps in the germination in case of peanut seed. Ethylene also helps in promoting root growth and root hair formation. Root growth and root hair formation. It initiates flowering and for synchronizing fruit set in pineapples. The last stress hormone is, or the plant growth regulator, which is come into the category of growth inhibitor, is abscisic acid. In 1963, Frederick T. Adicott and his co-worker identified abscisic acid. Abscisic acid was isolated from several abscission accelerating substances from cotton plant. As I earlier said, it is also called a stress hormone because it is generally synthesized in plant during the stress condition. Here is also the small change. Those are the physical effect of abscisic acid, not the cytokinin. Physiological effect of abscisic acid. Physiological effect of abscisic acid. It acts as a general plant growth inhibitor. It induces seed and bud dormancy. That means it causes seed and bud dormancy and inhibit seed germination. It stimulates the closure of stomata because due to the high temperature condition or the high temperature stress, the transpiration rate in plant increases and that causes the excessive loss of water. To avoid this, the abscisic acid stimulates the stomata to close. It plays an important role in seed development, maturation, and dormancy. Thank you.